Hello everyone, my name is Mary.
And my name is Kinga. We are molecular biology students, and today we will talk about the ethics of animal testing.
Since our field of study is heavily related to development of medicine and lab work, which is often worked on animals. In modern science, many important medical discoveries like vaccines and life-saving medicine have depended on animal testing.
For example, treatments for disease like cancer, diabetes, and infections were developed with the help of animal research. Because of that, animal testing has played a big role in saving human lives.
But at the same time, this brings up an important question. Is it right to use animals for scientific research and scientific progress?
Animals can feel pain, stress, and fear just like humans do.
And unlike humans, they cannot choose or give consent to be part of those experiments.
So we have a so difficult situation.
We want to continue scientific progress, but also want to treat animals in a fair and a human way.
To deal with this problem, scientists developed something called the 3Rs principle. This principle helps makes animal research more ethical and more responsible.
It doesn't completely stop the animal testing, but it tries to reduce harm as much as possible. In this section of our presentation, we are going to talk about what those 3Rs stand for, and they stand for replacement, reduction, and refinement.
These are three simple but very important ideas.
They help scientists think carefully before using animals, and they guide how experiments should be done and planned and done. First, scientists must ask, can we avoid using animals altogether?
Second, if we must use animals, can we use fewer of them?
And third, how can we reduce their pain and improve their conditions of life?
So the 3Rs are really about making better choices. They are not just moral suggestions. In many countries, they are part of the law.
This means the scientists are required to follow them. Before any experiments, they must be checked and approved.
Usually, special ethics committees review the experiments. They make sure that the rules are followed and that the animals are protected as much as possible.
The first principle that we will talk about is replacement, and this means avoiding using animals whenever possible.
Scientists always try to find out methods instead of using animals. Today, there are many alternatives.
First example is cell cultures. These are humans or animal cells grown in a lab, and they can be used to study how cells react to drugs or chemicals.
Another example is computer stimulation. Scientists can use powerful computers to model how the human bodies work, and this allows them to test ideas without harming any living.
There is a disadvantage to this method because it cannot show the long-term consequences on an organism with those chemicals that are put in the program. But there are also new technologies like organ-on-chip systems. These are small devices that copy how real human organs function. For example, there is a lung on a chip that can stimulate breathing,
or maybe a heart on a chip can show how heart cells respond to medicine.
There are also artificial skin models, and they are mostly used to test cosmetics and chemicals instead of using animals. And so instead of testing animals, scientists are trying to use something that behaves like human tissue, in this case skin.
Yes, the main idea is so simple. If we don't need animals, we should not use them. However, sometimes full replacement is still not possible.
And that is very obvious because living organisms and humans are very complex, and some processes can only be tested on whole organisms. Because then we can see how whole organism is as one reacting to a certain chemical or a certain medicine that we applied to it.
Yes. So, and the goal is to replace animal testing as much as possible in the future. The second principle that we are talking about is reduction. That means using fewer animals in experiments.
But this must be done meticulously. Scientists still need reliable and accurate results, so they plan experiments in advance very carefully.
They use statistics to decide the smallest number of animals needed. This helps avoiding using too many animals.
Another important method is sharing data. If scientists share their results, other scientists don't have to repeat those same exact experiments, and that prevents using unnecessary animals.
Also, modern technology allows more information from the experiment.
For example, imaging techniques can give a lot of data from a single experiment in a single animal.
This means fewer animals are needed overall, so reductions is about efficiency.
And also, it's about getting better results while using fewer animals.
The third principle is refinement.
This focuses on improving how animals are treated. If animals must be used, their suffering should be minimized. One way to do this is by using anesthesia if animals are going to keep living, or we should use painless ways to euthanize them.
This reduces or removes pain during procedures. Pain relief after experiments is also so important.
Another part of refinement is better living conditions. Animals should have enough space, proper food, and clean environment to reside in.
They should also have stimulations like toys or social interactions. This helps make reduce stress and improve their well-being.
Researchers are trained to handle the animals very, very carefully, and they learn how to reduce fear and avoid unnecessary harm to those animals.
Less invasive techniques are also used whenever possible. For example, instead of surgery, scientists might use imaging.
And the goal is very clear: to reduce pain, stress, and suffering as much as possible to those animals that we test on. Now we're going to talk about the ethical considerations of animal testing. Even with all of these improvements, animal testing is still widely debated. Some believe that it is absolutely necessary for medical progress, but
other believes it is wrong to use animals at all. Both sides have strong arguments.
On one hand, animal testing has helped save millions of human lives. On the other hand, animals cannot choose to participate since they're not conscious beings and they don't have a way to express their will.
And they can suffer during experiments. Because of this, strict rules exist.
Every experiment must be approved before it begins. Scientists must clearly explain why animals are needed.
They must also show that there is not better alternatives, and most importantly, the benefits must be greater than the harm.
So animal testing is only allowed when it's truly necessary.
These 3 R's help keep this balance between scientific progress and ethical responsibility.
Today, science is slowly moving towards less animal testing. New technologies are helping this change. For example, artificial intelligence that we are all aware about can predict how drugs will behave, but personally, in my opinion, it cannot fully replace the organic reaction that we get for a whole being and the whole organ system.
3D tissue models can act like a real human organs in some industries. Animal testing is already banned. For example, in cosmetics in many countries.
So this shows a clear direction in the future where science is going, and the science is becoming more ethical and more advanced at the same time. But the real question is, again, can those model organs truly replace the living organisms that are just naturally there and already have all the mechanisms to respond to chemicals, and can we truly replicate those in lab-grown organs and culture cells? In conclusion, the 3 R's are replacement, reduction, and refinement, and they are the key to the modern science today.
They help make research more humane and responsible. Even though the animal testing still exists, it is becoming more controlled and limited.
As science develops, ethics must stay very important and in the main focus, and the main question remains:
how can we continue to advance science while respecting all forms of life?